Assalamualaikum and hi, my name is Nur Nazatul Najwa binti Harunizam with metric number AN180176. I would like to present my proposal titled Cytotoxicity Synergy between Kelulut honey and sea cucumber extract on human cancer cell lines for development of potential anti-cancer supplement. Cancer is the world's second largest cause of death. It is becoming more common in all countries, developing and developed alike. As a result of the rapid development of cancer treatment resistance, as well as the severe toxicity and side effects of some cancer chemotherapy medications, there has been a surge in demand for new anti-cancer drugs, particularly from natural sources. Natural products, either structurally unmodified natural materials or synthetic derivatives, have played an essential role in the treatment of cancer for more than 40 years. Hence, natural substances with low toxicity is a good option. In this study, Kelulut honey and sea cucumber will be studied as both contain high nutritive value and valuable medicinal properties. Sea cucumber have leathery skin and gelatinous body shaped like a soft-body cucumber. The National Cancer Institute of the United States reported that 4% of marine species evaluated have anti-cancer characteristics from a survey of research publications published in the past 15 years. Due to the presence of bioactive agents, sea cucumber has been linked with therapeutic potential. These bioactive agents have cytotoxic action, apoptosis induction. They contain valuable nutrients such as vitamin A, magnesium, and calcium. Next is honey. Honey is a flavored viscous solution produced naturally by both honeybees and stingless bees. It is well known throughout the world for its high nutritious content and health benefits. In vitro and in vivo research have revealed its antibacterial, antiviral, antifungal, anti-cancer, antidiabetic, and antioxidant activities. Common honey producing bees in Malaysia include Heterotrigona itama, Apis dorsata, and Apis mellifera, which produce Kelulut, Joalang, and Acacia honey. Objective of this study is to analyze the physicochemical properties of Kelulut honey and sea cucumber extract. Second, to investigate cytotoxicity synergy of Kelulut and sea cucumber on human cancer cell lines. And last but not least is to determine cytotoxicity level on different human cancer cell types, which are human colon cell, human breast adenocarcinoma, human cervix adenocarcinoma. Next, I will proceed with chapter 2, which is literature review. First subtopic of literature review is about supplement. A nutritional supplement is defined as a product intended to supplement the diet that contains one or more dietary ingredients, including vitamin, mineral, herbs, amino acid, or other botanicals, and is taken by mouth as pill, capsule, tablet, or liquid according to the 1994 US Dietary Supplement Health and Education Act. When it comes to cancer, it's critical to remember that no dietary supplement can totally treat, cure, or prevent any type of cancer. Some supplements, on the other hand, may be able to help prevent cancer or aid in the cancer recovery. However, there is some problem and issue from previous study about the supplement. There is minimal evidence that multivitamins lower cancer risk from observational research, and there have been no randomized studies on multivitamins for cancer prevention. Therefore, observational studies in human populations may also be used to support a proposed association between a nutrient and a cancer outcome, rather than depending exclusively on in vitro and animal investigations. This evidence might help justify human experimentation, which is normally done in the form of randomized control trial, or we call as RCT. More dietary supplement randomized control trials are needed to determine their efficacy in cancer prevention. Next is consideration for selection of in vitro toxicity testing. Traditionally, animal models have been used to extrapolate to potentially harmful events in human in the toxicological evaluation of environment chemicals. While these animal models have offered helpful information on chemical safety, they are generally expensive, low throughput, and occasionally unreliable predictors of human biology and pathophysiology. Study found that in vitro give a more cost-effective alternative, and initially testing can be done with a larger number of sample or variables. Due to the complicated interconnected nodes of the cell signaling network, various altering methods must be applied to achieve therapeutic efficacy. Phytomedicines that exert therapeutic effect through the additive or synergistic actions of several chemical compounds acting at single or many target sites related to a physiological process can achieve this. The method is dependent on trial and error. If the effectiveness of the combination is more than the effect of either the agent alone or the sum of the effect of the individual agents, and antagonism is less than the expected effect of the combination, it is deemed synergistic. For testing synergistic effect, the isobol approach is the most convenient and experimentally acceptable method. For this combination, no studies on Kelulut and sea cucumber to treat cancer. However, one of the popular over-the-counter medicine in Malaysia to treat colds is Hurist cough syrup, uses the combination of honey and sea cucumber. Next is therapeutic effect of stingless bee honey. In comparison to honey bee honey, stingless bee honey has a higher moisture content, water activity, ash content, and free acidity, as well as a lower pH and total soluble solid content. Several factors influence the presence of certain ingredients in honey, including floral source, bee species, geographical origin, weather, harvesting season, processing method, and storage conditions. Several metrics, including moisture content, pH, free acidity, organic acid content, and 5-HMF content, are commonly employed to assess honey quality. As you can see, there is therapeutic effect of honey which include anti-cancer, antioxidant, and many more found from the previous study. Cytotoxicity activity of sea cucumber. Anti-cancer agents exhibit cytotoxic activity through blocking or preventing growth or mitosis of cancer cells. Sea cucumber compounds have been shown to have cytotoxic activity in various human tumor cell lines. For example, holoturin A3 and A4 generated by the Holoturia scabra species were found to be extremely cytotoxic. Hence, the anti-cancer activity of bioactive compounds derived from sea cucumber offers encouraging prospect for the treatment and prevention of human cancer. For physicochemical properties of Kelulut honey, moisture content, water activity, free acidity, and pH values of Kelulut honey from two different sources shown in the table. While for physicochemical properties of sea cucumber, collagen was extracted from sea cucumber using the PSC method and isolated using two alternative methods, which is dialysis and the ultrafiltration process. DPSC and UFPSC were the name given to the collagens obtained. Overall, the ultrafiltration approach for collagen isolation offered advantages over the dialysis method in terms of time efficiency, higher yield, and equivalent quality according to the data. Now, we proceed with chapter 4, which is methodology. I will use sea cucumber and Kelulut honey, which obtained from extracted ready-made commercial product. The sea cucumber samples were obtained from Selera Saurasa Foods, Sungai Petani Kedah, while Kelulut honey samples were obtained from Madu Kelulut Asli Rimba, Kahang Johor. Both of the natural substances have been extracted and in a liquid form. For synergistic testing, the main purpose of this study is to test the synergy of two agents, which are Kelulut honey and sea cucumber extract. The agents will be tested with different ratio of concentration to gain the best synergy. For each of the method that will be used, there will be different ratio combination of a sea cucumber extract and stingless bee honey. The ratio can be either the reduction of one or both honey and sea cucumber extract until the best cytotoxicity is obtained. For determination of physicochemical properties combination of Kelulut honey and sea cucumber, 4 parameters will observe, which is pH, water activity, moisture content, and cell density. For pH, a pH meter will be used to determine the pH of the sample. A pH probe will be used to measure the pH of 1 ml of the sample solution dissolved in 75 ml of distilled water at room temperature. All measurement will be carried out 3 times. While for water activity, the water activity of samples will be determined at room temperature using water activity meter. For moisture content, 5 grams of sample in stainless steel flat dish with tight fit cover will be heated at 105 degrees Celsius for 24 hours in a convection oven. Moisture content will be determined in unit of a gram per 100 gram. For the cell density, the trypan blue exclusion method, in which non-viable cells turn blue, was used to determine cell density. A sample of the cell suspension will be taken and diluted in trypan blue 1:1 in the Eppendorf tube, thus having a 2-time dilution factor. The mixture will be placed in a Neubauer chamber under an optimal microscope. The cell density will be defined by the following equation. For determination of cytotoxicity, the test will be performed by the MTT method. All cells will be routinely grown and maintained in Roswell Park Memorial Institute, medium with 10% fetal serum and 1% penicillin streptomycin. All cell lines will be incubated in a moisture-saturated atmosphere containing 5% CO2. To each well of the 96 well microplate containing 100 microliter of cell suspension will be added 100 microliter of extract in the concentration of 2.5, 5, 10, 20, and 40 microgram per liter. And the plate will then be incubated in a CO2 incubator at 37 degrees Celsius for 24 hours. After the addition of 100 microliter of MTT saline solution to each well of microplate, the plate will then be incubated for 4 hours under the same conditions in the CO2 incubator. Next, I will proceed with chapter 4 of this study, which is expected result. The aim of the project is to investigate the cytotoxicity of Kelulut honey and sea cucumber extract combination on human cancer lines. The toxicity of these agents will be evaluated using the MTT assay. The combination of both Kelulut honey and sea cucumber is expected to be toxic to human cancer lines, in which the calculation of IC50 value will show that the combination of Kelulut honey and sea cucumber extract has strong cytotoxicity against 3 different cell lines. The optimal concentration ratio and the cytotoxicity level of Kelulut honey and sea cucumber extract combination on 3 different cell lines will be determined. So as a conclusion, for this final year project 1, a research proposal has been submitted. The proposal consists of several chapters, including introduction, literature review, methodology, expected result, conclusion, and references. The overall aim of this study is to investigate cytotoxicity synergy between Kelulut honey and sea cucumber extract on human cancer lines for development of potential anti-cancer supplement. Cancer is a severe disease and a leading cause of death worldwide, as there are many evidence from published literatures on the medical benefits of Kelulut honey and sea cucumber individually. Combination of Kelulut honey and sea cucumber as a cancer preventive supplement has potential and promising. From the literature review exercise, many published study reporting on synergy of natural products, therapeutic effect of stingless bee, cytotoxicity activity of sea cucumber, physicochemical properties of Kelulut honey and sea cucumber are successfully recorded. Based on published study as the reference, the methodology for this study is proposed to achieve the set objectives. The lab experiment will be conducted during final year project 2 next semester. That's all from me. Thank you.